Hey everyone, welcome to the Exam Ignite. In today's video, we continue with the Module 2 of VTU subject Biology for Engineers BBOK407 4th semester under 2022 scheme. I have uploaded my Module 1 in my previous video. So in the Module 2, we will be focusing on real-life applications of the biomolecules. So these topics are very important and often appear in the model and previous year papers. I have segregated them according to the topic wise. Carbohydrates, in that we will study about cellulose based water filters, properties, advantages and disadvantages. PHA and PLA as bioplastics. Nucleic acids, in that DNA vaccine for rabies, DNA vaccine for COVID-19. Forensics, here DNA fingerprinting. Proteins, protein as a food, that is whey protein and meat analogous. Plant-based proteins. Then in lipids, biodiesel, cleansing agents, or you can say detergents. Enzymes, glucose oxidase in biosensors, lignolytic enzyme in biology. So I have referred the previous year question papers and model question paper 1 and model question paper 2. Based on that, I have added so in which year it was asked. Let us start. Introduction part. So in this chapter, we will be studying about carbohydrates, proteins, lipids, and nucleic acid. Carbohydrates, what is it made up of? It is built of what? That is sugar units, that is monosaccharides, disaccharides, and all. Function is what? Then we are having three main types. One is sugar, which is a simple carbohydrate. Then starch, which is a complex carbohydrate. Fiber, this is also complex carbohydrate. Special compound, that is we study about a cellulose. Here it is a complex carbohydrate, or you can say polysaccharide, which is made up of 3000 plus glucose units. This is renewable, biodegradable, and insoluble in water due to hydrogen bonds. What is the application of cellulose is? It is used for the water filters. The cellulose nanofibrils are used to create the bio-based membranes. So these membranes, these filters based on the cellulose pulp filters do usually have large pores, and that help in the percolation but need enhancement for the bacterial removal. That is, it has a large pores that filters the water, but the bacteria are not removed. To enhance this, what we use the methods. To enhance that, we incorporate antibacterial metal nanoparticles into that cellulose-based water filters. That is addition of silver and the copper nanoparticles for the antibacterial effects. Another one alternate method to the physically remove the bacteria from the water is using of positively charged filters to attract the bacteria, and it removes the negatively charged bacteria. Interesting, as no need of adding any toxic chemicals. In this method, we don't add any toxic chemicals. Next, key techniques that are used here is one is layer by layer modification is done. Then nitrogen analysis is done. Then scanning electron microscope. Then flow rate measurement. Then bacterial removal efficiency testing. Then the natural water sampling is filtered. Then fluorescence microscope is observed. Application of this is we use it in the cellulose filter, that is biodegradable, it is lightweight, it is efficient in removing the bacteria. Carbohydrate based plastics there are two types, one is PHA and PLA. We'll go into that. Actually, this is an important topic because it is asked in many model question papers and in the previous year question papers. Firstly, we have to know what is bioplastics. Bioplastics, it is derived from the renewable biological sources, that is starch or vegetable oil. What is it compostable? It biodegrades completely within how much? 180 days in the composting conditions. So based on that, bioplastics are of two types. One is bio-based and another one is biodegradable. Not all bio-based plastics are biodegradable, and not all biodegradable plastics are bio-based. You have to remember this. Bio-based is derived from the biological sources, that is, materials is on the origin of the carbon building block. Biodegradable means it can decompose through the action of microorganisms. This is the difference between those two. Now, what do you mean by PLA? Polylytic acid. It is what? It is a bio-based and biodegradable under the industrial composting. That is approximately you should have 58 degrees Celsius. What are the advantages? It should have good mechanical strength, high transparency and durability, easy to process, and it is non-toxic. What are the applications then? Packaging, biomedical devices, and in disposal utensils. So this is a structure of the PLA. You have to write the structures and diagrams in order to score in the biology. And mainly the notes is oriented of what are the main topics or the important topics that your answer should have, main highlighting points that your answer should have. You have to frame a sentence and you have to elaborate the points that I have given in the notes. Now, PLA life cycle, it's like from the photosynthesis, CO2 is absorption and the photosynthesis occurs. Then from the fermentation, extraction is done, processing is done, manufacturing of the PLA is done, then that gets biodegraded and that again in turn goes to the photosynthesis. This is how the PLA's life cycle is. Now, PHA. PHA it is 100% bio-based and biodegradable. It is produced by what? Bacteria through fermentation. So properties depend on it is type of bacteria, which type of bacteria it is, fermentation condition, substrates used. Advantages it is tunable, physical or mechanical properties. It can range from the brittle plastics to rubber like elastomers. You can make any of the plastic. It can replace the PP, PE, PS in the industries. This is the structure of the PHA. PHA life cycle is from the agriculture we get the bacteria or the biological waste that we do fermentation that gets to the biodegradable plastic. We do after it is used we collect the plastic. Either it will go to the industrial recycling or directly we do the anaerobic digestion, and with the biofertilizers we again put it into the agriculture field. Next topic comes the nucleic acid. Nucleic acid, DNA and RNA. Structure, it is made up of nucleotides. What are nucleotides made up of? It is made up of a nitrogen base that is A, T or U, C and G, a phosphate group and 5 carbon sugar that is ribose or deoxyribose. You can see the structure of the RNA, ribonucleic acid, and this is the structure of the D-ribonucleic acid, deoxyribonucleic acid. Polymer, DNA, sugar it has is deoxyribose, bases are adenine, thymine, cytosine and guanine. For the RNA, its sugar is ribose, adenine, uracil, cytosine and guanine. Functions is nucleic acid function is to store and transfer the genetic information. It guides the protein synthesis. Application of the nucleic acid is firstly DNA vaccine for rabies. So rabies is a preventable viral disease. Most often it is transmitted through the bite of the rabid animal, especially to the mammals. So the rabid virus infects the central nervous system of the mammals, ultimately causing a death in the brain or death. DNA vaccine uses PCL or NCO plasmid encoding the rabies virus glycoprotein gene administered to dogs to induce a long-lasting immunity. Advantages is no adverse effects of this vaccine, highly safe, effective prophylactic strategy for preventing the rabies. So the process, how it is done is gene from the immunogen it is taken, it is inserted in the gene of an expression plasmid, then they transfer the bacterial cells and grow the bacteria, purify the plasmid DNA, they immunize with the immunogen expression plasmid. RNA vaccine for COVID-19. Here it uses mRNA encoding SARS-CoV-2 spike protein. Example of the vaccine is Pfizer or Moderna vaccines. Working is mRNA enters the body cell. It instructs them to produce a spike protein so that immune system recognizes the spike protein and it builds the antibodies. mRNA does not enter the cell nucleus. Okay, main point. Another thing is it cannot alter the human DNA. I'll explain you. So this is a COVID-19 virus. It has a spike protein. mRNA with instruction for making the spike protein is developed in the lab. This one is developed. Now, that is what mRNA vaccine. When this is injected to a human body, that mRNA enters the cell, in the human cell. The COVID-19 virus spike proteins created. It is created. COVID-19 virus spike protein that is created in our cell. Now, antibodies are there. The spike proteins are recognized by the immune system of our body, which produces what? Specific antibodies against that COVID-19 vaccine. So for that, what? That immunity gets developed. If you are infected with the COVID-19 virus, antibodies bind to this virus and stop it from replicating. This is how we can cure. DNA fingerprinting in forensic. So it is also known as genetic fingerprinting or DNA profiling. What are the steps here is, firstly, we will collect the sample, that is hair, blood or skin. DNA extraction and purification is done, cutting the DNA with the restriction enzyme, then the gel electrophoresis, which separates the fragments. Then blotting, where DNA transferred to a nylon membrane. Radioactive probe or hybridization is done, and the X-ray detection. Application here is, we use it in the crime investigation, paternity test, identification in disasters. So I'll explain the process. Here DNA is extracted, restriction digestion is done, that is cut into fragments. It is put into the electrophoresis, that is expanded here. These are the wells. This is the largest bands and the smallest DNA bands. Then it is the DNA bands are transferred to the nylon membrane. Then the hybridization of the DNA is done, and to the X-ray sheets we see that. Next comes a protein. What are proteins? Proteins are the large biomolecules which are made up of what? One or more long chains of amino acid residues. These are found in muscle, bone, skin, hair, and all the body tissues. They form the enzymes, hemoglobin, hormones, and structural components of the cell. What are the functions of proteins then? They provide a wide range of vital functions, that is catalyze the metabolic reaction, enable the DNA replication, respond to the stimuli, that is to the hormones or receptors. They provide a structural support, that is collagen, transport the molecules, that is hemoglobin. Protein is what? It is a functional unit of genetic information execution. Protein structure and synthesis. So we are having four types of structure, that is primary structure, secondary structure, tertiary structure, and quaternary structure. Primary is sequence of amino acids that is dictated by the gene. Secondary is local folding of alpha helix or beta sheet. Tertiary is 3D folding driven by the side chains. Quaternary is multiple polypeptide chain combined. Now, and these are encoded by the gene, that is nucleotide sequence or the amino acid sequence. What is the genetic code is? Codons is 3 nucleotide sets. Each codon is equal to 1 amino acid. Translation process is there where the protein synthesis takes place. The mRNA is loaded into the ribosome. Codons on the mRNA match the anticodons of the tRNA. Translation RNA. tRNA delivers the amino acid assisted by what? Amino acetyl tRNA synthesis. Peptide bonds are formed. They grow nastic polypeptide chain, that is N terminus to C terminus. Importance of proteins in the cell. These are the chief cellular actors, that is executing the genetic information. Proteome, this is complete set of protein expressed in a cell is called. In E. coli, proteins are made up of what? Approximately 50% of the dry weight. DNA is around 3% and RNA is 20%. Protein as a food. So our body requires protein. Daily requirement is approximately 7 gram protein per day per 20 lbs or 9 kg body weight. What are the sources? We are having two sources. One is animal based and plant based. Animal based is complete. That is, we are having meat, fish, dairy, or eggs, all the essential amino acids. Here, actually, plant based are incomplete. That is, grains, pulses, nuts, seeds. They lack some of the amino acids. What do you mean by whey protein? It is a byproduct of the cheese. Okay, it's a byproduct of the cheese production. Main proteins present in the whey protein are alpha lactalbumin, beta lactalbumin, and serum albumin and immunoglobulins. These are the four things, main proteins you have to write in the exam. Notable component is glycomacropeptide, that is, which is not a protein, but it is present in the whey protein. Whey protein is commonly marketed as a protein supplement. This is processed by removing what lactose, lipids, and non-protein component. What is the role in the fitness? It has to be taken post the exercise. It enhances the protein synthesis. It promotes the muscle recovery, muscle building. Note that not effective when it is taken after the exercise. So I'll explain the steps how the whey protein is made. The cow's milk is shipped from the dairy farms. Okay, the milk arrives at the processing plant in the tanker trucks, where we do a during the cheese making process. Enzymes are added to separate the curd, and whey remains as a co-product of that. Now the liquid whey is pasteurized. After that, the whey is sent to the concentrated and purified. Here, two technologies can be used. One is membrane filtration or the ion exchange chromatography. After that, the final product is sprayed, dried into a powder. It is typically between the 80 to 90% protein. Now, meat analogs, it's a plant-based meat. What are they? Products that mimic the meat. Exactly, it mimics the meat in the texture, taste, appearance, and nutrition. It is made up of what? High moisture extraction from the plant proteins. What are the benefits? One, it is ethical, low fat and low calorie, low environmental impact, no animal diseases are carried, has been reduced. Ingredients used in this is one is soy protein, mycoprotein, soy legumulin. Plant-based sources, these are here protein extraction is done and the characterization. Formulation of the meat replica dough by adding the protein, fats or oils and binding agents, flavoring or coloring and some preservatives are added. And finally, processing of the plant-based meat analog using this machine. And finally, processed food is ready for distribution under the different labels. Plant-based proteins. So apart from this, where do we get? We get in the pulses, that is in chickpea, lentils, beans like in kidney, black or peas, other things like tofu, nuts, seeds, whole grains. Next, nutrition benefits is it is having high fiber, vitamins, minerals, helps the meat daily fiber intake to our body. Nutritional quality may be slightly lower than the animal proteins. What is the growing demand? Why? Because health benefits of the plant-based diets, it concerns over animal protein and the saturated fat. It is designed for sustainable food systems and ethical concerns, that is no animal welfare is done. View of protein has a positive nutrient. Next topic is lipids. Now, lipids are broad group of what hydrophobic or amphiphilic molecules. This includes fats, oils, waxes, steroids, phospholipids, fat soluble vitamins like A, D, E, K. Now, what are the functions? It is energy storage, structural component, that is cell membrane, it is phospholipid bilayer, hormonal signaling, then insulation and protection. Classification based on the ketoacyl and the isoprene subunits, they are classified into 8 types. Refer them. Now, bilayer formation is phospholipid is aqueous solution from lipid bilayers, basis of the biological membranes due to hydrophobic effect. That is why that forms the bilayer. Application is it is used in the cosmetic, food, and nanotechnology. Main application area it is used in the biodiesel production. Lipids from the food waste are used as a food stock for biodiesel. There are two methods. One is base catalyst and the biocatalyst. Base catalyst is KOH. It is 100% biodiesel in 2 hours. Biocatalyst, it is 90% diesel in 24 hours. Biodiesel is a long chain of alkyl esters, methyl or ethyl propyl or fatty acids. These are produced by what? Transesterification with alcohol. What are the sources for this is soybean oil, vegetable oil or animal fat. You can use anything. Advantages is these are compatible with standard diesel engines and can be used alone or blended with the gasoline. Also usable as heating oil. Another application is detergent, which we'll study next. This is the process of making the biodiesel, that is methanol and catalyst, oil and fat. These are sent to a transesterification unit or the separation. Here, washing and drying is done. We get the biodiesel. We also get a byproduct called glycerin. In detergents, membrane disruption, that is, detergents can bind to the hydrophobic ends of the phospholipid bilayer. This allows the solubility in water, breaking the cell membrane. Stages of solubility. Detergent binds to the lipid membrane. Membrane proteins are solubilized. Membrane ruptures occurs at the higher concentration. This is a detergent. This is a bilayer. This is how a mixed missile is formed. Next topic is enzyme, where enzyme biocatalysts. Here, proteins that speed up the chemical reaction by lowering activation energy. Types of enzymes, we are having 6 types, with each of them having different features or properties for them. Next, function, mechanism, it binds to substrate, forms the enzyme substrate complex, and finally releases the product. What are the applications of enzyme? Firstly is biosensors. Glucose oxidase in the glucose biosensors. Glucose oxidase is a widely used enzyme in the glucose biosensor due to the high stability and low cost. Here, GOX is a glucose oxidase. It catalyzes a redox reaction, transferring the electrons from the enzyme's active site to the electrode. Enzyme's active site to the electrode, enabling the measurement of the glucose level in the blood samples. An atmospheric glucose biosensor is created by immobilizing GOX on the electrode surface. During the enzymatic reaction, glucose is converted into D-glucono 1 and 5-lactonine. Oxygen is reduced by hydrogen peroxide. Now, the concentration of the glucose is qualified by detecting either the amount of the hydrogen peroxide produced or the amount of oxygen produced. Used in the decision detection, drug monitoring, and the pollution analysis. Analytes gets into the receptor where it forms an enzyme, and finally electrochemical reaction occurs, and that signal is sent to the amplifier. Next application is biobleaching. Here, lignolytic enzymes, they break down the lignin into the wood pulp. Includes enzymes like lacase, lignin peroxidase, manganese peroxidase, produced by the this biobleaching, it is produced by the white rot fungi for the second gen biofuels and eco-friendly paper bleaching. The process is given here. You can refer. This was about the module 2. Stay subscribed and stay tuned for the further updates and for the further modules. Thank you.